Welcome students, once again we're having a new video. In this case we will talk about a very interesting structure of cell known as cell membrane. Now some of you may remember that cell membrane has 4 or 5 main functions, which I'm going to try to summarize in here. But the most important of all will be that cells act like the body wall of the cell. Cell membrane acts like the body wall of the cell, so they're going to be the structures that are in charge of protecting what happens inside and outside the cell. So let's try to make a summary. Cell membranes isolate, in other words, means that this separates the inside from the outside. This will be the outside of the cell and this will be the inside. Cell membranes separate that, regulate the exchange of substances. Remember that cell membranes have a property known as selectively permeable capacity, which in other words means that they can control what comes in or what goes outside the cell. Also, chemical reactions. Some of the chemical reactions of the cell, like for example moving important ions like sodium or potassium needed for ATP production, will be in charge of cell membrane. The next one will be communication. Remember, one cell needs to be really in touch with another in order to know if they are part of the same body or they belong to the same tissue. And finally, transport. In other words, how substances are going to be moved. This point is directly related with the exchange of substance. The other thing you should remember is that cell membranes are always considered as a bilayer. In other words, it's like a sandwich, like I said in class, of two sides or two decks of molecules formed by phospholipids, which is the main component of cell membranes. But they are not only formed by phospholipids, they also have cholesterol, which is also another type of lipid, and then we will have proteins. And if you remember, I also said that there will be included a fourth type of molecule known as carbohydrates. These carbohydrates are going to be the one outside the cell in charging of reacting or identifying an external cell or external membrane with the actual cell that we are referring to. In other words, they are important identification tags for one cell to the other. So 4 biomolecules will form cell membranes: cholesterol, proteins, phospholipids, and carbs. These 4 are biomolecules. This is just another picture in which you can identify the different positions of the proteins inside the membrane and also the cholesterol. For example, we have this structure, which is an integral protein that is crossing all the bilayer of phospholipids. We have the peripheral proteins, which are the ones that are located just at one side of the cell membrane. It could be in touch with the extracellular fluid outside of it or with the cytoplasm inside. Now finally we will have cholesterol, which is a molecule that gives motility, a little sort of fluidity to the cell membrane. Now remember, another important concept is that you should remember that cell membranes are always defined as a fluid mosaic model. In other words, all these elements are going to be moving constantly. That's why they are defined like a fluid mosaic model. They are not stick together every time. They can be found at any position at any given time. Now, because cells are always surrounded with water, they have a solution outside it and also a solution inside it, in other words, the cytoplasm and the extracellular fluid. We need to know the physical properties of these molecules. Remember, they are molecules that are stick together to the physical and chemical laws of the bondings and all that stuff. Now, as you should remember, phospholipids, they have two properties. They have hydrophilic heads, which are these yellow guys over here, which loves water. In other words, they want to be near water. And we have hydrophobic tails, which can't stand to be near water. So once the phospholipids are together, they're going to take this disposition. Hydrophilic heads outside and inside the cell, and hydrophobic tails inside, just in the middle of the bilayer, or the like sandwich of phospholipids. Now let's try to remember some of the most important movements of the cell. Remember that the type of movement between or beneath the cell membrane that doesn't require energy is known as the passive transport. In other words, it's going to follow a gradient or a different concentration between these two regions, the outside and the inside, so the substance will be moving from where we have more concentration to where we have less concentration without using energy. This could happen in three different ways. Just crossing the membrane, and that's known as diffusion, using a protein that could be a channel or a carrier protein, which they do not use ATP as well. That's why we call it passive transport. And finally, the type of movement that pulls substances against a gradient. In other words, from we have less concentration to where we have more concentration, and also that requires energy, will be the one that we know as active transport. Now this is important because not all the substances are always going to be needed in the same concentration. Substance will be moved according to the cell needs. So remember, if the substance is moved from where we have less to where we have more, like in this case, this is known as active. If the other transport follows a gradient, will be known as passive transport. Now, if the cell is requiring to intake a lot of different particles at the same time, or expelling substances, a lot of different substances or particles at the same time, we will have two types of active transport that performs exactly what the cell needs. If the substances are getting inside the cell membrane, like we have in here, this process is known as endocytosis. Endo meaning inside the cell, okay? So the actual way in which these substances are going to move inside it will be using an organelle known as vesicles. If you remember that, vesicles are little bags of cell membranes that contain substances. So once the substances are inside the membrane is going to start like pulling inside the cell or towards the cytoplasm, the nucleus, and then the little vesicle will start formed once it's already closed. In the other hand, if the substances are needed to be expelled or outside the cell, this process is known as exocytosis, which is exactly the same but in an opposite way. The vesicle starts moving to the cell membrane. Once the vesicle is very near to it, cell membrane and vesicle will fuse, will stick together, they form just one structure, and the inside content of the vesicle will be expelled outside. Now, these two types of transport, remember, they are always known as active transport. In other words, they required energy. Finally, we need to remember that the movement of water between the outside and inside, or the exterior and interior of the cell, the movement of water, just water, has a particular name, a whole concept for it, known as osmosis. Now, this example is trying to remember, or try to remember to you, the three different types of solutions that we will have according to what is outside and inside the cell. We will have hypotonic solution, isotonic solution, and hypertonic solution. Now, these cells or these drawings represents red blood cells, the cells that we have running inside our blood, and they are very sensitive to different concentrations of water outside and inside the cell. So these arrows represents what it's going to be doing water, getting inside of it, getting outside of the cell, or also flowing constantly like in a little cycle. Now remember, the type of solution will determine how is going to be this water movement. Now let's try to make a comparison. Isotonic solutions, they always have this certain type of balance. The same amount of water and solids that we have outside of the cell will be exactly the same that we have inside. Now let's try to figure out what happens with hypotonic and hypertonic solutions. Now, for an hypotonic solution, what is going to happen is that the water is going to be found in a higher concentration outside the cell, meaning less solids, more water, and less solids. So the water is going to follow a gradient from where we found more to where we found less. And then the cell will swell. Remember that word. For the ones of you that don't know this meaning, swell means hincharse. Now, in an hypertonic solution, what is going to happen is exactly the opposite. Hypertonic solution has more water inside the cell at the beginning, and obviously water will follow a gradient, so it will leave the cell or exit the cell, leaving the cell shrinked. In other words, being little. For the translation, it means encogerse. Now remember, hypotonic solution will always cause the cell to swell. Hypertonic solution will always cause the cell to shrink. Now, this is not magic, remember, it always follows a gradient or the concentration of the substances, in this case, the solids and the solvent, which is water. Now, isotonic solution, which is most of the body fluids that we have inside, will have a constant movement, equilibrated movement between outside and inside the cell. So the cell has a normal shape. Normal shape. Okay, so I don't know what just I wrote over there, but remember the word was normal. Normal shape, okay? Now, for the ones of you that want to know a little bit more about this, I'm recommending you to follow these guys over here, the Amoeba sisters. They are a really cool like video bloggers, but they use these types of animations. You can find them all over YouTube, and they have very simple and interactive, some sort of funny videos to explain all the different biological processes we can have. So that's just a hint for you. Follow Amoeba Sisters on YouTube to try to find these GIFs, these videos all over the YouTube. So I hope you have enjoyed and liked also this summary about cell membrane. And well, we'll see you soon. Goodbye.